Hello. Actually, this is the first time I've done the uh, lecture online, so hopefully it all goes well. This is microbiology, chapter 9, and microbial genetics. Let's see how it goes. Okay, first we'll talk about plasmids. Actually, this is 139 slides. We'll go through them pretty quickly, and I'll tell you the ones that you don't really need to worry about. I've knocked out quite a few of them, but we should be able to go through them pretty quickly. So it talks about the uh, genetics in microbials, and so you see here it has plasmids. There's circular snippets, and this shows some microorganisms. They call it a mating dance between cells. They're connected by conjugation pili, which is where they actually transfer their DNA. Okay, we're scoping out the chapter. We're going to talk about chromosomes and genes. So they are packages of DNA. All cells are tightly wound. They wind their elongate strands of DNA into compact chromosomes. So the genes bearing specific codes are located at sites along the length of the chromosome. Okay. This is just as a show characterization of the DNA, which the deoxyribose backbone. Okay, so here we have DNA replication. So we're going to talk about the copying of the DNA blueprint. So the codes carried by DNA are conserved for offspring and future generations. This ensures that organisms will inherit the complete and accurate collections of genes that are required for survival. This blue part is the DNA, and they're connected, and these red parts is where each parent strand is replicated by uh, exact duplicate of the separated DNA strands. Then we're going to talk about transcription and translation. That's interpreting the blueprint that the DNA holds for. So DNA acts as a model for assembling RNA copies of its codes. So the RNA are transported to ribosomes to read the coded message and express it as useful cell proteins. So shorthand for that is it goes from the DNA to the RNA to the protein. And so this is this is messenger RNA that's in here. This is the one that's taken directly from the DNA. And these are transfer RNAs. This transfer RNA has a code or anti-code that relates directly to this messenger RNA. And this transfer RNA has amino acid at the top. Okay, we'll talk about that more in depth. Then we're going to talk about mutations. So mutations are changes in the genetic code. So like any blueprint or language, mistakes can change the meaning of the DNA message so that transcription and translation are unable to produce the intended protein. Has a variety of outcomes. Usually they're harmful. Sometimes mutations can be beneficial. Then we're going to talk about recombination. That's transmission of DNA from one cell to another. Some bacteria readily share their DNA codes with other bacteria and even share it with some viruses, as seen in this small vignette here. This results in bacteria with new genes and traits that help them adapt to changes in their environment. So this is like saying if one bacteria had viruses in it, and it actually takes some of the RNA or DNA from this bacteria, and if it infects another bacteria, it will inject that bacteria into the other. They will inject that RNA or DNA into the other bacteria. Okay, we're going to start with genetics and genes. First off, the genetics is the study of heredity. So the science of genetics explores the transmission of biological traits from parent to offspring and expression and variation of those traits, the structure and function of genetic material and how material changes. That's genetics and genes. So the levels of structure and function of the genome. So here's an organism level. So this is an organism. And then you go to the cellular level. And when one of these cells that are on this organism, at the cellular level, inside the cell, it has a nucleus. And inside the nucleus, you have chromosome. This is the chromosome level. And then making up this chromosome is the DNA. And it has the genes on it. So the genome is the sum total of genetic material or the DNA in the cell. So most exist as chromosomes for DNA. Some appear as non-chromosomal sites, like in the mitochondria, chloroplast, and plasmids. The genome of cells is DNA. Genome of viruses is DNA or RNA. So the chromosome, what is a chromosome? Chromosome is discrete cellular structure composed of a neatly packaged DNA molecule. Eukaryotic chromosomes are located in the nucleus and are multiple and they are linear. So eukaryotic chromosomes, they're in the nucleus and there's a lot of them and they're linear. Bacterial chromosomes are a single circular loop. Actually, all right, so then we have this is a cell that has, you can have a cell that has a nucleus, and you have chromosomes within the nucleus, nucleolus as well, mitochondria, chloroplast, and also you have plasmid in some fungi and protozoa. They have the circular DNA. Okay, then you have viruses. Well, actually, that's eukaryotic cells have the nucleus, and some of them have plasmids. Then you have prokaryotes, which has chromosome that's free floating. There's no organelles, no nucleus, and they can have plasmids as well, and free floating DNA in bacteria. Then you have viruses. What the viruses, I'm not sure why you can't see the bottom of that, but the viruses have DNA or RNA. So viruses are basically genetic material inside a protein coat and sometimes a capsule. So we'll talk about genotypes and phenotypes. So a chromosome is subdivided into genes, which is the fundamental unit of heredity responsible for a given trait. Those are genes. The site on the chromosome that provides information for a certain cell function. So a gene is a segment of DNA that contains the necessary code to make a protein or an RNA molecule. So three basic categories of genes. First, you can have genes that code for proteins, which are considered structural genes. Then you have genes that code for RNA specifically. Then you have genes that control gene expression, which are regulatory genes. So all types of genes constitute the genetic makeup. So genetic makeup is genotype. I need you to know genotype. All types of genes constitute the genetic makeup, which is called genotype. And then the expression of the genotype creates observable trait. So once the genetic makeup is expressed, it makes up the observable trait, the phenotype. Phenotype is how something looks. Okay. Okay, and the size of packaging of genomes. So here we have the smallest viruses are made up or have about, smallest viruses have 4 to 5 genes. And E. coli, which is bacteria, contains a single chromosome and it has 4,288 genes. Human cell has 46 chromosomes and they contain about 31,000 genes. Let's see. Okay, so the human cell has 46 chromosomes containing 31,000 genes. Okay, the packaging of DNA. The DNA molecule is compacted in the cell by supercoils or superhelices. So in prokaryotes, it's done by the action of an enzyme called DNA gyrase, which coils the chromosome into a tight bundle by a reversible series of twists into the DNA molecule. It's in prokaryotes. In eukaryotes, the packaging is more complex, with three or more levels of coiling, starting with a chain of nucleosomes, DNA around histone proteins. This just shows some of the, uh, like this is a eukaryotic cell. These are some of the histones, and the DNA coils around the histones. And then when it's time to actually express the DNA, then this DNA will uncoil, and then it will have loosely condensed chromosome, and then it will actually show as chromosome, intact chromosomes. So the structure of DNA, it's a double helix. So basic unit of DNA structure is the nucleotide. So it's a deoxyribose sugar, it's a phosphate group, and it's a nitrogenous base. The nitrogenous base is either adenine, guanine, thymine, or cytosine. Those are the nitrogenous bases that will make up DNA. So nucleotides covalently bond to form a sugar phosphate backbone. What I need you to know is I need you to know the nitrogenous bases that we have for DNA. Adenine, guanine, thymine, cytosine. Adenine always pairs with thymine. Guanine always pairs with cytosine. So here's what I mean by saying they pair. This is one side of the DNA molecule. This is another side of the DNA molecule. This is the backbone. On the one side, it has adenine. It will bond with thymine on the other side. Adenine and thymine will always pair. And this one has cytosine. It will always pair with guanine on the other side. This is guanine, cytosine. These are what we call base pairs, and they always match up. Adenine with thymine, guanine with cytosine in DNA. So nitrogenous bases covalent bond from the 1' carbon of each molecule and span the center of the molecule to pair with a complementary base on the other strand. So you have adenine pairs with thymine, has 2 hydrogen bonds, and then guanine pairs with cytosine, 3 hydrogen bonds. This just shows the double helix. This is when it's all convoluted around the histones, and this is as it's opening up, and as it opens up, and it opens up, and you can see on one side it has base pairs, or it has bases on one side, and the complementary bases on the other. Adenine with diamine, cytosine with guanine, thymine with adenine, guanine with cytosine. So what they have is they have what they call an anti-parallel arrangement. So on one strand, the helix runs 5' to 3', and the other side is oriented 3' to 5'. They're opposite because the pairs have to be opposite each other. Each strand provides a template for the exact copying of a new strand. So the order of bases constitutes the DNA code. And it's just saying 5' goes down to 3'. That means that this side has to be 5', goes up to 3', because the base pairs are opposite each other. Guanine pairs with the cytosine, thymine, adenine, cytosine, guanine, adenine, thymine. So if this one's moving this direction down this way, this one has to move this direction the opposite way because it's the opposite base pair. So maintenance, the significance of DNA structure. So maintenance of code during reproduction. So consistency The constancy of base pairing guarantees that the code will be retained. So when strands are separated, each strand serves as a template for replication of the molecule into an exact copy. It also provides variety. The order of bases responsible for RNA and protein synthesis, thus for the phenotype of each organism. So you have a variety of bases that's responsible for RNA and protein synthesis. So here's a concept check. In DNA, adenine is a complementary base for what? And cytosine is complementary base for what? Of these, the answer is adenine pairs with thymine, cytosine pairs with guanine. Let's see. I need you to know adenine, there's four bases: adenine, thymine, guanine, and cytosine. And I need you to know that adenine pairs with thymine, and then cytosine pairs with guanine. Okay. Overall replication process. Replication occurs in both strands simultaneously. So when it separates the two strands, both sides will replicate. So it's called the semi-conservative process. So the parent DNA molecule is uncoiled, so it unwinds and separates the two. The two strands are separated, exposing the nucleotide sequence to serve as templates. That's what it's going to be built on. A template is what bases are going to be built on. So two new complementary strands are synthesized by using each single-stranded template as a pattern. So basically all that's saying is you have the helix that's all wound out, and it's a double helix, and that's in the blue. And then when it separates, the blue serves as a template, and it's going to produce a new red strand, single strand. So each side has its own red single strand. So all chromosomes have a specific origin of replication. The site as the place where replication will be initiated, where we'll start. The origin of replication is AT, which is adenine thymine rich. So thus less energy is required to separate the two strands. There are two replication forks where new DNA is being synthesized, each containing its own set of replication enzymes. So here's where it shows the figure of the DNA replication. And you see with this one, it is when this DNA unwinds, then this one line becomes a template for this RNA, and then this is a line for a template as well, but it has to go in the opposite direction. And so they both go from the 3' to the 5' side. From 3' to 5'. So when it goes this way, this one goes this way. This one goes from 3' to 5', but it has to go the opposite direction. So it has to replicate in or be transcribed in the opposite direction. And so in segments. So the chromosome to be replicated is unwound by a helicase. Know that it's a helicase. It's an enzyme that unwinds the DNA. That enzyme that unwinds the DNA is called a helicase. Forms a replication fork with two template strands. So the template for the leading strand, which is blue, is oriented 3' to 5'. This allows the DNA polymerase 3 to add nucleotides in the 5' to 3' direction toward the replication fork, so it can be synthesized as a continuous strand. And so let's look at this. So all it's saying is this can go from the it's replicated from the 5' to 3'. I mean, from 3' to the 5'. And so it's being replicated in the 5' to 3' direction. So it's uninterrupted. That's the initial strand. Okay. Then this is the lagging strand. It still is being replicated from the 5' to 3', but it has to do it in segments. So the template for the lagging strand runs 5' to 3'. That's opposite of the lead strand. So to make the new strand in the 5' to 3' orientation, synthesis must proceed backward, away from the replication fork. So before synthesis of the lagging strand can start, a primase adds an RNA primer to direct the DNA polymerase III. The synthesis produces unlinked segments of RNA primer and new DNA called Ozaki fragments. So let's look back at this. This is that RNA primer, this little piece here. This is the RNA primer here. So the red is a new strand. This little part here is the RNA primer. These are called the DNA polymerase III. And this that unzips it is the enzyme that uncoils or unzips the DNA. That's called what? Called helicase. And so you can look at the key down here. That's the helicase. It has RNA primer. It's that little green part at the beginning. The new strand is red. So this is a new strand. And new strands here. And then the original template strand is the blue strand. It's the original template strand. And then you have a primase that actually starts the replication on the lagging strand. Then you have the DNA polymerase III. So the DNA polymerase III causes making the new RNA strand on both sides. And then DNA polymerase I. DNA polymerase I actually connects the two strands that are separate the Ozaki fragments. And you have ligase. And we'll talk about that. The ligase actually connects the two Ozaki fragments. That's where you have DNA polymerase I removes the RNA primers and fills in the correct complementary DNA nucleotides at the open sites. And then finally, the unjoined ends of the nucleotides must be connected by a ligase. So here we talk about more specifically, I want you to know helicase. Helicase unwinds and unzips the DNA double helix. So helicase. And the RNA primer is synthesized at the origin of replication by a primase. And then DNA polymerase III adds nucleotides in the 5' to 3' direction. So the leading strand is synthesized continuously in 5' to 3' direction. And then the lagging strand is synthesized 5' to 3' in short segments. So the overall direction is 3' to 5'. Okay, so DNA polymerase I removes the RNA primers and replaces them with DNA. But I want you to know here, I want you to know definitely helicase, and then also the DNA polymerase III is the one that adds nucleotides in a 5' to 3' direction. So on the leading strand, it's continuous. On the lagging strand, it's in short segments. And those short segments are called Ozaki fragments. So this is where it says it has two replication forks, and it actually replicates both ways, this way and that way. So when the replication forks meet, the ligases link the DNA fragments along the lagging strand. So the replication proceeds, One newly synthesized strand loops down, and when the forks have gone full circle, termination site shuts replication down. So the two circular daughter molecules remain connected briefly, but are nicked and separated by a helicase. And this will show a picture of that. This is the two replication forks, and it creates a brand new DNA. And so it actually ends up being connected. It's connected here, but then it nicks it so that it separates the two. And so then you have two daughter cells that are produced. Okay, so this is showing the enzymes and what they do. And so I want you to know helicase, and then the primase, synthesized RNA primer, DNA polymerase adds new bases to new DNA chain, proofreads for mistakes. DNA polymerase 1 removes RNA primers. Ligase is the final binding of nicks in DNA during synthesis or repair. Gyrase helps supercoiling, it wraps it back up. Okay, so know this slide. Helicase, primase, DNA polymerase 3, DNA polymerase 1, ligase, and gyrase. So, concept check. Why must the lagging strand of DNA be replicated in short pieces? So look at each of these. Look at each of these. Are these what are true, false? Why must the lagging strand of DNA be replicated in short pieces? True or false? Is it because of limited space? No. Is it to avoid distortion of the helix? No. Is it because the DNA polymerase works in only one direction? And to make proofreading of the code easier? No. So the DNA polymerase works in only one direction. Okay, that's applications of genetic code. So genetic information in DNA molecules is conveyed to RNA through the process of transcription. Genetic information in DNA is conveyed to RNA through the process of transcription. Know what transcription is. Information contained in the RNA molecule is then used to produce proteins in a process called translation. So a wide variety of specialized RNAs act by regulating gene function. Definitely know transcription and translation. So here, let's look at this picture. This is a DNA that we're starting with. You have transcription of DNA, and that forms messenger RNA. Look for transcription of DNA. And it will also form transfer RNA and ribosomal RNA. So transcription, you'll form the different types of RNA. But there's different functions for each of these. You have ribosomal RNA, which will make up this ribosome, which will hold the other RNAs to make protein. You have transfer RNA, which actually has anticodon at the bottom, which is a sequence of 3 bases. And then you'll have amino acid on the top that's associated with these 3 bases. Then you'll have the messenger RNA, which has bases as well, but everything's based in 3s, triplet code. We'll talk about that in a few minutes. So that's transcription, that's making the RNA. And then you have translation. That's where messenger RNA pairs up with, let's say the codon in the messenger RNA pairs up with an individual transfer RNA that has an anticodon with 3 bases, and associated with each individual transfer RNA is an amino acid. So the amino acids combine to form a peptide bond, and it adds to the length of the polypeptide or the protein. The DNA also has regulatory RNAs. I mean, when it produces RNAs, it has regulatory RNAs, which are double-stranded RNAs and single-stranded RNAs. And then different regulatory RNAs will regulate or stop transcription or translation or encourage it, and also will stop the production of RNAs at the DNA. So different types of RNAs, I want you to know the different types of RNAs that are produced. Messenger RNA, which messenger RNA will have codons. You have transfer RNAs, which will have anticodons. You have ribosomal RNAs, and then you have regulatory RNAs. Okay. So the gene-protein connection, how do you get from a gene to a protein? So each structural gene is an ordered sequence of nucleotides that codes for a protein's primary structure. So groups of 3 consecutive bases, which are called triplets, or codons, on one DNA strand are transcribed into RNA sequence triplets. Those are codons. Each triplet of nucleotides on the RNA specifies a particular amino acid. So a protein's primary structure, or chain of amino acids, determines its shape and functions. Proteins contribute to cell phenotype. Phenotype is how something looks. It has enzymes and structural proteins. What this is saying is that nucleotides, when you have, let's say the nucleotides are adenine, guanine, cytosine, thymine. And so when you have adenine, guanine, cytosine, thymine, guanine, cytosine, it can just go along, but it's split up in threes. For every 3, then you have a 3 here will have corresponding nucleotide on the copy of this strand of DNA. And on this copy, it have this 3, and then this 3 nucleotides are associated with a specific amino acid. So whatever the code is on the DNA, you have the opposite code on the messenger RNA, and then it's transferred to transfer RNA has an amino acid with the opposite code of the codon. It has an anticodon on the transfer RNA, and on the other side of the transfer RNA, it has the amino acid. Okay. So the general structure of the ribonucleic acid, or the RNA, is different than that of DNA in several ways. First off, the RNA is a single-stranded molecule that can assume secondary and tertiary levels of complexity, leading to specialized forms of RNA. So that's where we have the messenger RNA, transfer RNA, and the ribosomal RNA. So the RNA contains uracil instead of thymine. Remember with the DNA, we said we have adenine, thymine, guanine, cytosine. So RNA has uracil, but no thymine. We said that thymine paired with adenine. So now, if there's no thymine, the uracil will pair with the adenine. So it contains no thymine like DNA as a complementary base pairing mate for adenine. That means that uracil in RNA will now pair with adenine. Then, of course, the sugar in ribose is in RNA is ribose rather than deoxyribose. So here's a table that kind of describes messenger RNA, transfer RNA, ribosomal RNA, and then also primers. Messenger RNA is translated Okay, the others are not translated. Okay, so the messenger RNA carries the DNA master code to the ribosomes. So the messenger RNA is the one that interacts directly with the DNA. Transfer RNA carries the amino acids to the ribosomes during translation. So the transfer RNA has amino acids on one side, and it has the anticodon on the other. Then the ribosome RNA forms a major part of ribosomes and participates in protein synthesis. Only one of these that is translated is the messenger RNA. So the transcribed version of a structural gene are genes in DNA. That's the messenger RNA is the transcribed version of a structural gene or genes. Know that the messenger RNAs are the transcribed version. They're synthesized following the complementary base pairing by process similar to synthesis of leading strands during DNA replication. So the message is in triplets called codons. Know that messenger RNAs have triplets called codons. So the messenger RNA, short piece of messenger RNA, illustrates the general structure of the RNA. Single-strandedness, repeating phosphate ribose sugar backbone with single nitrogenous base, and uses uracil instead of thymine. So here's the nucleotide bases. And so you have guanine, cytosine, uracil, and adenine, cytosine, guanine. Each one of the triplets will actually ultimately represent amino acid. So you look at there's how many triplets are there in this line. And so you count up the triplets, you don't necessarily count up the nucleotide bases. So then we have transfer RNA. The transfer RNA acts as a translator of the messenger RNA code into a protein. So the bottom loop of the clover leaf that it creates exposes the transfer RNA specific anticodon. So remember the messenger RNA had codon, transfer RNA has anticodon, and it's complementary to the messenger RNA codon. Okay? So then it also has binding site for amino acids that's specific for each anticodon. Let's see. So with this, it shows that this is the anticodon. So this is the transfer RNA. And so the anticodon has this 3-triplet pair that's going to pair with the codon on the messenger RNA. And these 3 nucleotide bases correspond to an amino acid that attaches on the other end. Okay, then we have the ribosomal RNA. Let's know that the ribosome is made up of an RNA as well, ribosomal RNA. So all this blue is the ribosomal RNA. This linear nucleotide bases forms the messenger RNA. This is what actually took the code directly from the DNA. This one here. It attaches to one part of the ribosomal RNA. Then a transfer RNA will come and have nucleotide bases, a triplet nucleotide base that will actually correspond to a triplet codon on the messenger RNA. And this triplet nucleotide base is called the anticodon on the transfer RNA. And so the transfer RNA has these 3 bases, and those 3 bases are associated with a specific amino acid. So this one has 3 bases associated with a certain amino acid. Okay, so this is how the different types of RNA correspond. Ribosomal RNA is blue, that kind of holds them all together. Messenger RNA carries the codon, which is directly from the DNA, and then you have the tRNA, which actually has the anticodon and it has amino acid. So during transcription, an RNA molecule is synthesized using the codes on the DNA as a guide or a template. I'm not going to require that you know the different stages. Just know that transcription produces messenger RNA from the DNA. So we won't go through each of the stages. Okay, major events of transcription. We won't go through the major events. Just know that with transcription, the messenger RNA is copied from the DNA. Then we have translation, which is the second stage of gene expression. Now, all elements needed to synthesize protein, which are messenger RNA, transfer RNA, and the amino acids, are brought together on the ribosomes. So this is where translation occurs. The process occurs in 5 stages. You have initiation, elongation, termination, and protein folding and processing. So here, once again, all of the elements needed to synthesize the protein. You have the messenger RNA, you have the transfer RNAs, and you have the amino acids. This is where they all come together. So master genetic code is represented by messenger RNA codon, and then their specific amino acids that they'll correspond to. This code is universal among organisms and it's redundant, which means different codons code for specific amino acids, and more than one codon can code for amino acid. So what this says is that if you have cytosine, uracil, uracil as a codon, then the amino acid will always be leucine. If you have cytosine, guanine, cytosine as a codon, the amino acid will always be arginine. And then there's certain codes, like you have uracil, guanine, adenine. There is no amino acid that's corresponding with it, and so it's a stop codon. And that's the same with uracil, adenine, adenine, uracil, adenine, guanine. That's a stop codon. And then there's a start codon, is known as adenine, uracil, guanine. And so it's associated with methionine as an amino acid, and that's a start codon. By the way, each of these names is an amino acid. All these are amino acids. These are names of amino acids. The ones that have the names here. Right? So that's amino acids. And these are the code, the triplet codes that codon that will actually produce this amino acid. Okay. So transcription, which produces messenger RNA that's complementary to the DNA code. So during translation, the transfer RNAs use their anticodon to interpret the messenger RNA codons and bring in the specific amino acids. It's important to know this slide, to understand this slide, that the messenger RNA is produced, like you have a messenger RNA that's produced from this DNA, messenger RNA produced, and then from the messenger RNA, it has corresponding transfer RNAs, and the transfer RNA has a corresponding amino acid. don't have to worry about this part in translation. Um, so this what happens in translation. This is the messenger RNA comes along and it has the codon. It has a start codon. So it has the anticodon that's associated with the transfer RNA. That transfer RNA has an amino acid on it. And then right next to it, you have another codon, anticodon pairing. And this amino acid this transfer RNA has an amino acid that's associated with it. When these two are locked in this ribosome, then it brings these amino acids close together and they form a peptide bond. So this where it says the second transfer RNA with complementary anticodon fills the A site. This is where a peptide bond is formed between the amino acids of these neighboring transfer RNAs. So these are in here because of these codon anticodon communication, and therefore the amino acids that are associated are close to each other and form peptide bonds. Then the first tRNA leaves, the next tRNA goes into the next site, which is called the P site. But now it has the two amino acids because it made a peptide bond from that first one. And then the line will move along this ribosome will move along the line of this messenger RNA to grab the next codon. And it will fill the space with another tRNA that will correspond with that codon. That's where we have this. The next codon, transfer RNA will come in that corresponds to that codon, and it has its associated amino acid, and it will lock in, and then this amino acid will connect with this amino acid, form a peptide bond, and you'll have a chain of 3. And that's where this, once it comes in, and this anticodon and codon meet up, then this amino acid that's associated with this transfer RNA will form a peptide bond, and will start getting longer. What's going to happen is this transfer RNA can now leave because these two are bound to this one. And that's what happens. This TNR leaves. This one now has 3 peptide bonds or 3 amino acids. It will go over to the P area, and a new transfer RNA will come into the A segment and will correspond to this codon. That's the next codon in the messenger RNA. And then it just continues on. It forms a peptide bond, and now you have a sequence of 4 amino acids. This transfer RNA will leave. This one will jump over to, this whole ribosome will move along messenger RNA. So this one will jump over into this, and then the next one will be associated with AUC. So then ultimately you get to a termination codon, which is UAA, UAG, UGA, and there's no corresponding transfer RNA for those, and they don't have a corresponding amino acid. So when that codon is reached, ribosome falls off, and the last tRNA is removed from the polypeptide. So concept check. The transfer RNA is the molecule that does what? Does it contribute to the structure of the ribosomes? No. Well, that's true, false. No, that's false. Does it act in the transcription of DNA? Well, the transcription is from the DNA to messenger RNA. No, that's false. transfer the DNA code to the messenger RNA? No. Well, does it act as a translator of the messenger RNA code into a protein? Yes, because the transfer RNA has the anticodon and it has a amino acid. So D is correct. So you have polyribosomal complex. This is where a lot of ribosomes will actually be on one messenger RNA line, and it will make a lot of proteins all at once, and then they'll join. So you have eukaryotic transcription and translation. So eukaryotic translation and transcription do not occur simultaneously. So transcription occurs in the nucleus and translation occurs in the cytoplasm. So the eukaryotic start codon is AUG, but it does not use formal methionine. So it is, the start codon is AUG, but does not use methionine. Eukaryotic messenger RNA encodes a single protein, unlike bacterial messenger RNA, which encodes many. So eukaryotic DNA contains introns, which are intervening sequences of non-coding DNA, which have to be spliced out of the final messenger RNA transcript. We'll talk about introns in a couple minutes, but I definitely want you to know introns. Okay, so eukaryotes' gene coding sequence are exons. So the gene coding sequences in eukaryotes are called exons. You know that. And then they're interrupted by segments called introns. Introns are non-gene coding sequences in eukaryotes. So introns are non-coding, so they are transcribed, but they're not translated. So introns are transcribed, which means they form messenger RNA, but they're not translated. So that means they do not have transfer RNAs. So they are removed before translation. So this type of splicing does not occur in prokaryotes. So on this, you have the DNA template, and you see this section is an exon. This section in green is an intron. So you have when the mRNA is made from this DNA, you have everything intact. You have the exon and the introns. But then you get what they call spliceosomes, which splices out the introns. These introns are the non-coding portions. They're spliced out. And then the rest of these exons are pieced back together. And this is the messenger RNA. And this messenger RNA is now ready to be used to be translated, because these exons are spliced together, and now this can actually be used for translation. And once again, in eukaryotic cells, all of this occurs in the nucleus, and translation occurs in the cytoplasm. So regulation of protein synthesis and metabolism. Genes are regulated to be active only when their products are required. So in prokaryotes, this regulation is coordinated by operons, which is a set of genes, all of which are regulated as a single unit. Two types of operons. So inducible operons, the operon is turned on by substrate. So if the substrate, which the substrate is something that is going to be acted on, they have catabolic operons. So those are enzymes that are needed to metabolize a nutrient are produced when needed. Remember, catabolism is breaking something down. And so when you metabolize or break down a nutrient. So they're produced when you need nutrients broken down. Then you have repressible operons. Those are genes in a series that are turned off by the products synthesized. So they're an anabolic operon. So the enzymes used to synthesize an amino acid, they stop being produced when they are not needed. So once you make enough, then it turns it off. It's inducible and repressible operons. We won't have to worry about lack operon, lack operon, lack operon, lack operon. We won't get into the arginine operon. Okay, mutations. Let's talk about mutations. Mutations are changes in the genetic code, a change in phenotype due to a change in genotype. Where genotype is the gene, phenotype is how it looks. But a change in the phenotype due to the change in the genotype is called a mutation. So a natural, non-muted characteristic is known as a wild type. So something that's a natural, non-muted characteristic is known as a wild type. So an organism that has a mutation is a mutant strain showing variants in morphology, nutritional characteristics, genetic control mechanisms, resistance to chemicals, etc. That's a mutant strain. Wild type is a non-mutated. So isolating mutants. So first off, culture is exposed to a mutagen. So whatever that mutagen is, it could be a chemical, it could be X-ray, it could be ultraviolet light, but a culture of a substance is exposed to something that will mutate their genes. So isolated colonies are transferred on a master plate. So tiny clump of cells are picked up and transferred to a plate with complete medium and another with an incomplete medium. We'll see if a mutation allows some of them to grow differently with different media. So colonies in a complete medium plate, missing from the incomplete one, are mutant colonies that can be subcultured for further use. So colonies in the complete medium that are missing from the incomplete one are mutant colonies that can be subcultured for further use. And this is just the description how they do it. Um, they actually have the culture, and they have the two plates that are one is uh, incomplete and one has everything that they need to grow, and they just see which one. All strains grow, mutant colony does not grow, and one that had a missing portion. And then whichever one, uh, then they reculture from colonies. So you have causes mutations. You can have spontaneous mutations, so random change in the DNA due to errors in replication that occur without a known cause. That's a spontaneous mutation. No spontaneous mutation, no induced mutation. So do know those. So spontaneous mutation, random change in DNA, and we don't know what the cause is. Induced mutation that results from exposure to known mutagens. So you can have physical, primarily radiation, or chemical agents that interact with DNA in a disruptive manner. So here's some selected mutagenic agents, their effects. I'm not going to ask you this. So here's some categories. You have a point mutation, which you have added, deletion, or substitution of a few bases. Missense mutation causes change in a single amino acid. So one amino acid, that's a missense mutation. Nonsense mutation changes a normal codon into a stop codon. Remember I said there's a few codons that have the stop feature? So if you change a normal one to a stop one, silent mutation alters a base but does not change the amino acid. Then you have a back mutation when the mutated gene reverses to its original base composition. Then you have a frame shift mutation when the reading frame of the messenger RNA is altered. Definitely know this slide and the different categories of mutations. So you have the classification of major types of mutations. First, you start off with a wild type, non-mutated sequence. An example of a non-mutated sequence, the big bad cat ate the fat red, and actually should be bug. It should be a G there. The wild type sequence of a gene is the DNA sequence found in most organisms and generally considered the normal sequence. So this is, since it's in threes, they just say, like, just for example's sake, they're saying, like, if you had these threes set up like this, each one of these represents a nucleotide base, and they're in the threes. So these are the codon threes, and then how it affects the final protein. Okay. So the categories of mutations based on type of DNA alteration. So if you have substitution mutations, so if you have missense, remember a missense mutation causes a different amino acid to be incorporated into a protein. So the big fat cat, right? So the F, the fat, is changed to M, so that's one base, which that means it's going to change this amino acid. So missense mutation causes a different amino acid to be incorporated into a protein. So this is a protein, but it's going to have a different amino acid right there. Effects range from unnoticeable to severe based on how the new amino acids alters protein function. But it's just that one amino acid that changes. So that's the missense. Then if you have a nonsense mutation converts a codon to a stop codon. So it's supposed to be the big bad fat cat, da 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 da. But if you mistakenly had a mutation that made a stop codon, it's like resulting in premature termination of protein synthesis. So this protein that's been synthesized, it stops prematurely. The effects of this type of mutation are almost always severe. Okay? Then you have inversion mutations. Inversion arises when adjacent letters, so letters right next to each other, uh, exchange places, which alters one or two bases depending on the location of the inversion. This does not change the reading frame, but can result in significant changes in amino acids and protein functions. So like these amino acids next to each other exchange. Remember, it should be the big fat cat ate the fat, the inversion, the big fat cat. So it should be BIG. This G should be here, and this B should be there. The big fat cat ate the red bug. So this inversion mutations right next to each other, they actually switch over. Then you have frame shift mutations. So you can either frame shift, you can add a letter or delete a letter, and it causes a change in the reading frame. So it reads these three, the big, and then so it should be bad. And so what they did is they added a B, so this B that should be here is now into the next one. So big, bad. So instead of the D, it has a B. It makes a different amino acid here, but it also makes different amino acids from that point on. Because this, instead of having CAT, it's DCA. Okay? And the deletion, same type of thing. The big, and it should be bad. So BAD would be the next three. The B is gone. So it goes AD and captures the next C. So it's going to be a completely different amino acid. So it causes a change in the reading frame of the messenger RNA, resulting in a protein in which every amino acid after mutation can be affected. Because of this, frameshift mutations almost always result in a non-functional protein. Okay? The repair of mutations. So since mutations can be potentially fatal, the cell has several enzymatic repair mechanisms in place to find and repair damaged DNA. So you have DNA polymerase, which proofreads nucleotides during DNA replication. You have mismatch repair, locates and repairs mismatched nitrogen bases that were not repaired by the DNA polymerase. And then you have light repair for UV light damage. We have excision repair, locates and repairs incorrect sequence by removing a segment of the DNA and then adding the correct nucleotides. So the Ames test. Any chemical capable of mutating bacterial DNA can similarly mutate mammalian DNA. So any chemical capable of mutating bacterial DNA can mutate mammal DNA. So agricultural, industrial, medicinal compounds are screened using the Ames test. So they have an indicator organism, which is a mutant strain of Salmonella that has lost the ability to synthesize histidine. So it's Salmonella that doesn't make you sick. This mutation is highly susceptible to back mutation, which means it can go back to being an infectious type. And with the Ames test, you actually have a control plate and a test plate, and you just see uh, when you expose one of them to mutagen, how it affects the sample. So you have positive and negative effects of mutations. Mutations lead to non-functional proteins are harmful and possibly fatal. So mutations that leading to non-functional proteins, they can be harmful, possibly fatal. So organisms with mutations that are beneficial in their environment can readily adapt, survive, and reproduce. These mutations are the basis of change in populations. That's a good point to know. Let's see if I can find a test question on that. So organisms that do have mutations that are beneficial, they can readily adapt, survive, and reproduce. They're the basis of change in populations. Any change that confers an advantage during selection pressure will be retained by that population. So which of the following mutations would cause a frame shift mutation? The silent mutation. Silent means it don't see any change. Missense mutation. Nonsense mutation. If you deleted something, would it cause a frame shift mutation? Yes. Deletion mutation. So you have genetic recombination that occurs when an organism acquires and expresses genes that originated in another organism. That's genetic recombination. So you have three means for genetic recombination in bacteria. You have conjugation, transformation, and transduction. Conjugation as transfer of a plasmid or chromosomal fragment from a donor cell to a recipient cell via direct contact. So an example is a gram-negative cell donor has a fertility plasmid, which is F factor, that allows for the synthesis of a conjugative pilus. So donor, which is F positive cell, transfers fertility plasmid through the pilus to recipient, the F minus cell, which becomes an F positive cell. Here shows an example of it. This is the F factor, and so this one does not have it, and so does not have this factor. One that does, they have a pilus that makes contact. It teaches it how to actually um, develop this pilus, and so both of them now have the F factor. High frequency recombination, donor's fertility plasmid is integrated into the bacterial chromosome. So when that conjugation occurs, a portion of the chromosome and a portion of the fertility plasmid are transferred to the recipient. And this is transfer of the F factor. Once again, you have this one does not have it, this one has it, and it physically transfers it over. Now both of them have it, that factor. They basically teach each other. And this is high frequency transfer, involves where it's transferring, it has a F factor integrated into its DNA or into its chromosome. It actually injects that portion, into a recipient, and now the recipient has it, that gets incorporated into its chromosome. And so it forms two separate cells that both now have the F factor. So chromosome fragments from a lysed cell means it's burst, are accepted by a recipient cell, and the genetic code of the DNA fragment is acquired by the recipient. So donor and recipient cells can be unrelated. So you have two bacterial cells, and one gets exposed to the DNA of another one, and that could be transferred. They don't have to be related. So useful tool in recombinant DNA technology. So what this is saying is that you can just have some DNA floating around, and a bacterial cell can actually ingest it, and it actually gets incorporated in. Now that bacterial cell incorporates it in the DNA, and from that point on, it has that characteristic that was just floating freely. It's just kind of describing that. So what's interesting with that is you can have a situation where you have a situation where you have a strain of colony that they're live strain, and the effect is that it will kill its host. So now if you have a live R strain and it gets infected, and the host will survive. So the host is not affected by the R strain. The host is affected by the S strain. What's interesting is this. So if you heat kill the S strain, that's the one that would kill it. If you heat kill this S strain, the host will survive. It's injected with it. But now if you have heat killed S strain, but then you have a live R strain. Remember, R strain did not cause the host to die. And you have killed S strain, these two come together. The result is that it will kill the host. So in essence, from the heat killed S strain, the live R strain took that knowledge on how to kill from these. and it ends up killing the host. So what ends up happening is you have live S and R strains isolated from the dead mouse. So you ended up with S strain, live S strain, and live R strain. Um, so transduction, bacteriophage. Bacteriophage are viruses that infect bacteria. So bacteriophage, which are viruses that infect bacteria, serve as a carrier of DNA from a donor cell to recipient cell. So two different bacteria can actually get transferred DNA from one to the other via a virus. So there's two types: generalized transduction, that's random fragments of disintegrating host DNA are picked up by the phage or the virus during assembly. Any gene can be transmitted this way. Then you have specialized transduction. A highly specific part of the host genome is regularly incorporated into the virus. A known portion of one bacterial DNA is incorporated into the DNA of the virus. That's specialized transduction. And it just talks about generalized and specialized transduction. Da, da, da. So this is the virus that infects the bacteria. So this is the viral DNA, and this is the host DNA. So the host DNA and the viral DNA take over a portion of the host DNA and make new viruses. But when it makes new viruses, a portion of the host DNA gets incorporated in with the viral DNA. The virus takes over the cell, it lyses the cell, and so now the viruses go out to infect other cells. When it infects other cells, when it injects it, it injects not only its own DNA, but a portion of this bacterial DNA into the new bacteria. And so now this bacteria now has a portion of a different bacterial's DNA. All right. And so this is the same type of thing. It's just that this um, the prophage within the bacterial chromosome. So this is the prophage within the bacterial chromosome. It breaks off that DNA but it contains some of the bacterial DNA. It makes new viruses. The new viruses break out, and now you have both. Um, it has both the DNA of the virus and the other bacteria, and now it'll actually produce some bacteria that has all of it incorporated in, some of it that has the this original host bacterial DNA that is separated out from the other bacterial DNA. I'm not going to get that deep in this with you. I won't ask a question on that. I won't ask about transposons. Uh, so let's go to genetics of animal viruses. So viral genome, that's the genetic makeup in the virus. One or more pieces of DNA or RNA that contain only genes needed for production of new viruses. So the virus will infect a host cell and inject their genetic code that is specific for making more viruses. Then we have viruses require access to the host cell's genetics and metabolic machinery to instruct the host cell to synthesize new viral particles. Most viruses contain normal double-stranded DNA or single-stranded RNA, but other patterns exist. So, with few exceptions, replication of the viral DNA occurs in the nucleus. The genome of most RNA viruses is replicated in the cytoplasm. But in all viruses, the viral messenger RNA is translated into viral proteins on host cell ribosomes using the host transfer RNA. The viral RNA is translated into viral proteins on the host cell ribosome using the host RNA. And this is just a picture of the replication of double-stranded DNA viruses and how they go about doing it. This is replication of single-stranded RNA viruses. Uh, all right, that's a lot of slides, but I've jotted down quite a few different parts where I've actually said know this. So 2, 4, 6, 8, 10, 12, 14, 16. So I haven't made up the test yet, but I'm going to gear it towards the things I said to know during this presentation. So do well. I will talk to you all soon. All right, thanks.